Once that primer is laid down, DNA polymerase can extend the sequence by adding deoxyribonucleotides, synthesizing DNA. DNA polymerase III has the capacity to synthesize a very long strand at a time, meaning it's continuous. It doesn't go on and off. On the other hand, primase synthesizes short segments. You get about 11 nucleotide primers. DNA polymerase III, on the other hand, can go on for an immense number of nucleotides, making it highly processive. In the 5' to 3' direction, it synthesizes a complement to the parental strand. The parental strand DNA, oriented 3' to 5', is the leading strand, shown on the bottom here. Once the leading strand is started, it continues with DNA synthesis on that strand until it reaches some sort of block. It has highly processive, continuous synthesis for very long stretches. Note that there's an inherent asymmetry in this process. The growing strand and the template are oriented in opposite directions. We add a nucleoside triphosphate to the 3' end of the strand, and we synthesize 5' to 3' with the template oriented 3' to 5'. When the nucleoside triphosphate is added, it releases pyrophosphate, and we're ready for the next nucleoside triphosphate. DNA polymerase III is very fast, synthesizing 5' to 3', with helicase opening up DNA as we go along. DNA synthesis is only 5' to 3'. So what do we do about the lagging strand? Because the replication fork is continuously opening and exposing new DNA on this strand that is anti-parallel to the leading strand. How is that newly opened DNA primed? The solution is discontinuous synthesis. On the leading strand, DNA primase synthesizes the primer at the origin of replication specifically. But for the lagging strand, it recognizes the replication fork and lays down multiple primers along the lagging strand. DNA polymerase can extend each of the primers. DNA polymerase I, which is distinct from DNA polymerase III, extends the RNA primer with DNA nucleotides. Though DNA polymerase III can also sometimes be used for longer stretches of DNA synthesis on the lagging strand. Remember that we're laying down and extending primers successively